Morning all. If you've been hearing some conversation, it is because we have lost our physician, our MPN physician for this morning, for this morning's event. Dr. Paul had an emergency with his little boy and they are in the ER. The child is going to be okay. He had an allergic reaction to something, but he just sent me the most incredible photo of the little guy hooked up to some stuff. But anyway, so bear with us. What I'd like to make sure that you know is that we are going to focus on, our industry partners, obviously giving their presentations. Nora, could you please hide your screen in your video? Thank you. And then we'll go through each presentation, but after each presentation, the physicians or patients or whoever has registered for this event may ask questions of that organization through us. So you just post your question and we will be happy to ask it. And so if you hear something or see something that you're interested in, please let us know. Or if you have questions about that, please let us know. We have done this. We're trying to do 2 of these per year, and we have found them to be extremely useful to both patients and physicians. Yesterday was focused on just the physician or just the patient, I'm sorry. And it was fantastic, and everyone had so much to share, and it was really great. And so I look forward to some robust conversation today as well. I'd also like to point out to everyone that today is MPN Awareness Day. And I'm especially happy to say this because I was part of a coalition years ago that decided that this date would be the day of MPN Awareness Day. And it has proven to be a pretty wonderful tribute to the MPN community. So many people do so many different things across the country and in other countries on this date. If you want to bring it to anyone's attention that, either for your patients or for others in the community, today's a good day to do it. Obviously, September is National Blood Cancer Awareness Month as well. Yesterday, we talked about COVID shots, the booster shot and flu shots, and the consensus was definitely to get the booster shot and to get vaccinated, first of all, if you haven't as a MPN patient. And secondly, to definitely get the booster shot when it's available to you. It is important, and with the Delta variant and other variants that are now pretty pervasive or becoming pervasive, it's important that you take care of yourself and stay safe. And the question was asked, what about the flu shot and getting a booster shot? And basically, yes, it's a good thing. You should do it. Perhaps you could keep a week in between those 2 injections, but definitely get your flu shot as well. And this is just information that we gave to the patients, and this program is being recorded as well. So when we send this out to our database, it will reach many, many more people than will be participating today. And so without further ado, I think most of you know us. I'm Ann Brazo. I've been in this space for 17 years, first with the MPN Research Foundation in Chicago for almost 9 years, and love them as partners. They do a great job in research and cutting edge research, fantastic work. And I was very proud of the work we did there. And I started MPN Advocacy Education International 8 years ago. Kathleen Michael's been with me pretty much ever since then. Actually, it's been almost 9 years because she came a little later. And Mallory Tim is now with us, who's fantastic. And she really has worked hard to make sure these Zoom calls or Zoom webinars work and happen. So I want to thank them for today and for all of the work that they do. So moving on, our first speaker, our first industry partner speaker, I would like to say, just an overall thank you very much to all of our partners. We would not ever be able to do what we do without their support. All of the organizations that work in the MPN space get support from our partners, most of them that I know of. And we are not unique in that way, but we have long-term relationships with many folks here today. And I'm just overwhelmed, like I said yesterday, very overwhelmed by what is happening in this space. I mean, when I started, there were maybe 3 to 5 people that were very into MPNs, and maybe a couple companies here and there. And it was my job to establish relationships with those folks. Now it's been an incredible, talk about proliferation, just, a huge number of organizations and companies that are interested in MPNs, new drugs in the pipeline. It's just incredible. And I'm so excited about all of it because our patients need your help and they need new treatment options. So our first speaker is from Insight Corporation, who has supported us for years, and we thank them very much. And Dr. Peg Squire is the Group VP for the U.S. Medical Affairs team at Insight, and she will be presenting first. And so please welcome Peg. Hi, Peg.
Hey. Hi, and thank you, Anne, and thanks so much to all of you. I'll go ahead and share my screen so that you can see the slides. All right, and as Anne mentioned, these will be recorded, so you'll have a chance to access details later. Thank you again for having me today, and I'm the head of U.S. Medical Affairs at Insight. It's my honor to present the agents that we currently have in full clinical development for myelofibrosis. All right. So I'll be talking about 5 compounds today. 4 of them are investigational. And so, of course, that means that there is no guarantee that any of them will be proven safe and efficacious and that they will come to the market. The 5th, of course, is ruxolitinib, and that is a marketed compound. This is its U.S. indication statement for MPNs for patients with myelofibrosis and polycythemia vera. So Insight has a deep and longstanding commitment to research in MPNs and in myelofibrosis. We seek to address the underlying pathophysiology of the disease, and we also want to address the individual needs of patients throughout their own treatment path. We think about that in terms of their line of therapy. So you can see that depicted across the slide here as they have their first line therapy. As that first line therapy potentially starts to wane in efficacy, or potentially they don't have the optimal effect of the therapy. And then as they go on to need additional lines. We also think about individual patient populations within each line of therapy, as you can see depicted from top to bottom. So if there are subsets within each line, so patients who have anemia or thrombocytopenia, and we seek to be able to provide different treatments for different patient populations and different clinical trials that fit within that. So we'll be discussing each of these compounds, each of these trials in the subsequent slides. The first focus of my short presentation here is on PI3 kinase delta, and I'd like to start by telling you why. Now, of course, myelofibrosis is driven by hyperactivation of JAK signaling. And so here you see JAK2 in its habitat in the cartoon, and you also see PI3 kinase delta. PI3 kinase delta is the predominant PI3 kinase isoform in MPN cells. It's known to be active. It's known to be one of the primary ways that JAK2 signals cell proliferation. And so because it's downstream, we think it is one potential resistance mechanism for JAK2 activation in MF. That got us interested in potentially testing a PI3 kinase delta in the suboptimal setting. We know that JAK inhibitors and PI3 kinase delta inhibitors show preclinical synergy, and we showed using our inhibitor parsaclisib, which is a next generation, highly selective PI3 kinase delta inhibitor, that we have clinical activity. So we see effect on spleen and symptoms in patients who have suboptimal response to ruxolitinib on its own. That trial, our phase 2 trial called 201, has shown interim results most recently presented at EHA in June, and actually this week at the SOHO meeting, which is a virtual meeting. So based on the phase 2 results, we've launched 2 large global phase 3 trials. This is the first, it's called LIMBER 304. We use the acronym LIMBER throughout our trials. It's our aspirational acronym. It means leading in MPNs beyond ruxolitinib. So this is parsaclisib, our PI3 kinase delta inhibitor, added on to ruxolitinib in the suboptimal setting. So here patients come on, they've received ruxolitinib for at least 3 months with a stable dose of at least 8 weeks. They have to have splenomegaly and symptoms. We've tried to make that as broad as possible. So splenomegaly of at least 5 centimeters below the left costal margin, symptoms with a total symptom score of 10, with the 7 symptom scale scored from 1 to 10 for suboptimal response. And they continue on the ruxolitinib dose that they were previously treated on, and they receive either parsaclisib at 5 milligrams q.day, which is a relatively low dose, or placebo. Patients who receive placebo do have the ability to receive crossover after the assessment for the primary endpoint, which we hope will make the trial more attractive for patients and for sites. And we have other factors in the trial which we hope will facilitate enrollment, including allowing patients with platelets down to 50, as well as intermediate 1 and 2 and high-risk patients by DIPS. So again, that trial is open and it's a large and global trial. Okay, excuse me 1 second. I just have to start presenting again as my presentation froze.
And I had a question for you, Peg.
Sure.
So in talking about placebo and crossover, so my understanding of crossover is when the placebo, when there are some changes in that patient with placebo, and then a crossover. Can you explain that, please?
Sorry, say that again to allow.
Can you explain, placebo to crossover to actually get drug?
Sure. So for this trial, patients who are randomized onto the study and are randomly selected to receive the placebo instead of the study drug, after they are assessed for the primary endpoint, so they've crossed 24 weeks of participation, they are allowed to become unblinded and find out if they received active study drug or not. If they didn't receive the active study drug, they're allowed to cross over and then receive the active study drug.
Thank you.
Does that answer the question?
It does, thank you.
Okay. And can you all see my slides now?
Yes.
Okay, great. All right. So now I'm talking about the other trial, which is the upfront trial. So this is a very similar study to 304, which was suboptimal. This is frontline. This is for JAK inhibitor naive patients. So again, similarly, patients are randomized to receive either ruxolitinib plus parsaclisib or ruxolitinib plus placebo. Again, platelets as low as 50 will make a patient or are an eligibility criterion for the trial. Patients who are intermediate 1, 2, or high risk may be eligible. And then the symptom and spleen thresholds are similarly broad for this patient population. There is also a provision for crossover on this trial as well. And again, this is a large global phase 3 study. Moving on to the rest of our portfolio, itacitinib immediate release. This is a new formulation for us. Itacitinib is a JAK inhibitor that we've studied previously in sustained form. In that form, it was basically only a JAK1 inhibitor, but in immediate release form, it also has some activity against JAK2. And we think this may hit a sweet spot for some patients who need some activity against JAK2, but who may have some considerations about cytopenias.
Excuse me 1 second, Peg. Your slides did not advance. So.
Oh, they didn't?
No, your slide didn't advance.
Okay, let me try that again.
It should be in presentation mode.
It is, yep. Let me just test it. Okay, there. Okay. The fun of a new.
Yes, I see the next.
How's that?
Yeah, there you go.
Okay. All right. So we see itacitinib IR, Limber 213.
Right.
Okay. So this again is our JAK inhibitor that inhibits JAK1 and a little bit of JAK2. The trial is in the second line setting. It's after ruxolitinib and or fedratinib. Again, we think that this may help patients who could use some JAK1 inhibition and a bit of JAK2 inhibition. Platelets can be as low as 50 for intermediate 1, 2, and high risk patients. All right, I'm just going to check. Do you see Limber 103?
Yes.
Excellent. Now we're cooking with gas. Okay, so this is our BET inhibitor, 57643. And I know at least one of my colleagues is going to be talking more about BET inhibition later. So I'll just make it short. BET inhibition is very interesting in MPNs. BET inhibitors are transcriptional regulators that attach to acetylated chromatin. We think that inhibition may have effects on anemia, spleen, symptoms, and other parameters of disease. This is our BET inhibitor, which is in phase 1/2. And so this trial is in dose escalation for patients with MF, MDS, and overlap syndrome, and also includes dose expansion in monotherapy and in combination with ruxolitinib. This trial will be in a more limited footprint, but of course, we hope to expand into larger trials very soon. And then the last trial, and please do stop me if you don't see it, is Limber 104. And this is for our ALK2 inhibitor, the 928 compound. And again, I know that at least one of my colleagues will be discussing ALK2 inhibition a little bit later, but suffice it to say that this target is very compelling in myelofibrosis because of its potential to regulate hepcidin, which regulates iron levels and can affect anemia in myelofibrosis. So we are studying it here in monotherapy and in combination with ruxolitinib. This is for patients in the second line who have anemia. And we are again doing dose escalation and then moving on to study it in expansion, both as monotherapy and with Rux. Okay, and you'll have this in recording, so you'll be able to access the details. Right. And then finally, I want to thank you for your attention. And if you want to learn more about any of the studies for possible participation in these or future studies, or if you think you have a patient who may qualify for one of the trials, this is all of our contact information by phone, by email, or our website, insightclinicaltrials.com. Thank you all very much.
That was excellent. Thank you very much. And we did talk a lot yesterday about combination therapies. And so I think it's great that you have other drugs in the pipeline that could, address anemia and some other issues that MPN patients have that are taking Rux. So thanks, Peg. It was great to see you. If we get another question during this hour, so we'll call on you, okay?
Thanks, Ann. I'll stay tuned.
Okay, thank you very much. So moving on, I'd like to introduce Dr. Hugh Reinhoff, who's the CEO of Imago Biosciences. We've known Hugh and his staff for quite a few years now as well. And we're anxious to hear what's going on with their drug. And Hugh, thank you for joining us.
My pleasure. Can everybody hear me?
Yes.
Okay, super. Thanks a lot. I'm calling in from San Francisco, where Imago is based. And as Ann says, appreciate the opportunity to speak and also to support the organization. We're big fans of that. So I am the founder and up until very recently served as kind of the chief medical officer of Imago. So I'm happy to present this. Actually, Wanjian Hong has joined us from Genentech as the CMO. She presented yesterday. She's relatively new to the company, so she's asked me to do this. I'm happy to do that. So we have spent the last 9 years putting together a program focused on lysine-specific demethylase. We've been in clinical trials in MDS, AML, myelofibrosis, and ET. And what I'd like to talk to you today about is mostly ET and myelofibrosis secondarily. And I have something similar to what Peg has shown, which is our drug bomedemstat, or IMG 7289, is an investigational agent that's not approved for use in the United States, but it is undergoing global clinical trials for a number of myeloid neoplasias, including MF and ET. So this is a snapshot of the company. We have an ongoing ET study, which is currently enrolling in a number of sites, I think 25, 26 sites around the world. And I encourage people to identify patients who have failed the standard of care to enroll in this study. We also have a myelofibrosis study, which is fully enrolled and should have its sort of first readout towards the end of the year. That's when all the patients will have reached 24 weeks. Not sure when we'll have all that data, but sometime either at the end of the year or early next year. And that's exciting and will be a prelude to further studies. We also have under development and will soon dose a combination study similar to what Peg described for ruxolitinib with other agents. I don't know how many of these studies there are going on, but there are a lot, and they all have their own rationales. We think that in a patient population that has a suboptimal response to ruxolitinib, that bomedemstat added to that regimen will provide a lot of the benefits of bomedemstat, which I'll get into, without a lot of the downside that you can often bring in with the second agent. So it's tolerability and an additional clinical benefits. We have a polycythemia vera study going on at a single site, and we'd like to expand that. We think there is a place for bomedemstat for the treatment of P. vera, and we can talk a little bit more about that later. We also have two programs focused on hemoglobinopathies and solid tumors. Both of these are new chemical entities that interact with LSD1 in a completely distinct way in which bomedemstat does. And I'll get into that a little bit. So this is our basic paradigm of how we think about myeloproliferative neoplasms in the context of lysine-specific demethylase, which is an enzyme that works in the nucleus, regulating hematopoiesis principally. actually at every stage. So in the early stages of hematopoiesis, you see the stem cell using LSD1 to balance self-renewal and proliferation. And in particular, the malignant stem cells that have acquired AML or MPN mutations, we're all familiar with those. Those have a greater dependency on LSD1. And in animal models, which we've published and presented in a number of forums, those LSD1 inhibition helps to extinguish the malignant stem cell population, which we think actually can lead to potentially exhausting that population and forestalling the progression of the disease itself, but also to AML, which, as you know, is a lethal outcome in this disease for the most part.
Excuse me one moment. You need to share your screen so we can see your slides.
Oh, I'm sorry.
And that's down at the bottom.
Hold on there for a sec. Got to end the show. Okay, hold on there for a sec. Really sorry about that.
That's okay.
I thought that was being done automatically. Yeah, hold on there for a sec.
Down at the bottom of your screen.
Yeah, I see. I
Yeah.
just got to get my.
Okay, sorry.
No, it's fine. Let me see if I can find it. Let's see. Hold on for a sec. No, why can't I see that? Let's see. Can you see this?
Yeah, and then, yes, and then I
Okay.
see all of them. There you go. That's
Okay,
it. That's
great.
it.
Okay.
Perfect.
I'll just quickly go over this again, just so you get to see it. ET, our study is half enrolled, and we'd like to complete that enrollment by the end of the year or early next year. So I encourage people to find sites for that. There are 25 across the globe. This myelofibrosis study, phase 2, is completely enrolled, and we should get the readout towards the end of this year, early next. And this is the myelofibrosis study that I was discussing, similar to what Peg has described with Insight's compounds, similar rationale with respect to suboptimal RUX response in patients, adding bomedemstat for its additional clinical benefits and its safety and tolerability profile, which I think is very compelling. So this is the slide that I was talking about earlier, where LSD1 is critical for the self-renewal properties of stem cells and malignant stem cells, those that carry the mutations associated with AML and MPNs, are more dependent on LSD1 activity and hence are more susceptible to LSD1 inhibition. And we do see in our patient populations, this is both in AML, ET, and in MF and hopefully in ET. We see a decrease in those mutant allele frequencies. That's JAK2, ASXL1, EZH2, all the mutations you'd like to see go down. And the clinical correlates of that is progression to AML. And I'll get into that. LSD1 is also required later on in differentiation for the maturation of megakaryocytes. And in ET, for sure, the activated megakaryocyte is really the soul of the disease. This is the cell that makes platelets, obviously. It also makes many growth factors that remodel the bone marrow and myelofibrosis in particular, and is one of the sources of inflammatory cytokines, which give rise to the constitutional symptoms that characterize both ET and MF. So by targeting this cell specifically, we can reduce the platelet count. And in fact, we use the platelet count in both of our studies as a biomarker of bomedemstat activity on megakaryopoiesis and thrombopoiesis. So in our preclinical models, and this is a JAK2 mouse model, as long as we treated these animals with bomedemstat on a daily basis, they survived, whereas the littermates who were treated with control or vehicle eventually succumb to the disease as patients do, particularly in myelofibrosis. So bomedemstat is an irreversible inhibitor of LSD1, which we discovered back in 2014. As I said, it's been in a number of patients now, more than 160 actually. And as I said, we do personalized dosing by starting with a fixed dose for all patients, ET and MF, but then titrate platelet counts down to a target range. And this, this turns out to be a convenient way to maximize exposure to the drug, but also minimize any of the untoward effects that might come from bomedemstat. And that's principally actually in the setting of these diseases, it's the effect on platelets. So platelets being the biomarker is also the endpoint, obviously, in ET. So we can titrate to the endpoint in that disease. And we have a good regulatory tailwind here for its prosecution for approval here in the United States and in Europe. So our ET study is, as I said, an international study. It's an open label, once daily bomedemstat. These are in patients who are high risk in need of cytoreduction, which is almost half the patient population. But we're focused on those who are intolerant or resistant to a standard of care, which in the United States, as you know, is mostly hydroxyurea. Although there are some in other countries, it could be interferon, it could be anagrelide. But the vast majority of patients in our study are actually hydroxyurea resistant or intolerant. So the objectives of the study are standard. There's safety and tolerability, which we think is a really important part of the profile of bomedemstat. It's obviously, in the case of ET, to reduce the platelets to less than 400,000 and also the white count into the normal range, obviously in the absence of thromboembolic events and progression of disease. And as I said earlier, we do monitor the frequency of 261 genes that are recurrently mutated in the myeloid malignancies, including MPL-CALR, JAK2, ASXL1, et cetera. And we follow those serially throughout the course of treatment to see what impact we have on those frequencies, but also to identify any new mutations that might arise, which would contribute to progression to AML in many cases. So this is using the platelets as the primary endpoint. You can see all these patients had a platelet count above the normal range. When they're washed out, they typically have a rebound in their platelets. And they, as I said, start out at a fixed dose. And you can see within a couple of weeks, these patients, their platelet counts come down. And typically, they require either a little dose titration up or down, depending on the exuberance of the response. And once they're on a stable dose, they typically stay within this range. And thus far, about 83% of the patients have achieved this platelet count endpoint with 6 or more weeks of treatment. Very encouraging results in a patient population that is either intolerant or resistant to other standards of care. And with respect to the white count, we see something similar. We see a little bit of a rebound in many of the patients, and some of them sort of declare themselves as having leukocytosis in about maybe a third or a quarter of these patients. And once again, on that same regimen that brings platelets down into the normal range, we also see the same with white count. And the white count, as you know, is an independent risk factor for thrombosis and is a required endpoint for the FDA. Importantly, we're not seeing an impact on erythropoiesis. So this just makes the point that bomedemstat, or LSD1 inhibition in our hands, is not a general myelosuppressive agent. And these patients, despite the wide range of baseline hemoglobins, they're all stable throughout the course of this disease using these doses. So we're very encouraged with ET that we have a safe and tolerable agent thus far that's able to bring most of the patients, 80+% of the patients into the normal range, bringing the white count to heal without having a negative impact on erythropoiesis. And with respect to symptoms, this is not a particularly symptomatic patient population compared to MF, but they certainly do have symptoms. And in those who had symptom scores over 10, the majority of those patients had some improvement in their symptoms. So they get relief on that side as well. Just switching gears a little bit with a few minutes that I have left, we also have this myelofibrosis patient population under study. As I said, it's fully enrolled, and we're putting together this data as it rolls out to us. A very similar strategy, open label once daily bomedemstat. These are patients who have platelets over 100,000, and we titrate that bomedemstat down to a platelet count of 50,000 and leave them at that. And the endpoints are very similar to other studies you've seen: symptom score reduction and spleen volume reduction. Importantly, if you look at the demographics of this patient population that has failed the standard of care, and that would be ruxolitinib and in some cases fedratinib, this is a very heavily treated patient population, been in many of these studies that you're familiar with, but most importantly, they have a very high mutation burden that inclines them to transformation to AML, and that's something we're studying very closely in this patient population. And just very briefly, we see improvements in symptom scores in these patients, and we also see a decrease in spleen volume. We admitted patients into this study who had relatively small spleens, because many of these patients actually do have significant symptoms and require some kind of treatment. And the bigger the spleen in our hands, the more it falls. So I think, this will be a secondary endpoint in our phase 3 studies. So I'll just summarize and say, we've got an irreversible inhibitor of LSD1 in this program in the form of bomedemstat. We also have reversibles and other allosteric inhibitors, which we're using for other indications. We've treated a number of patients and have a good safety database on tolerability and AEs. Remarkably, in the 4 years we've done the myelofibrosis study, we've only had 10 SAEs, and these were all one-offs, and so we don't have any serious concerns about that. And I would say that going forward, we plan to do a phase 3 study in ET next year, as soon as we complete this study, and we will attempt to do the same thing in myelofibrosis as soon as that study comes to fruition. And I'll leave it at that.
Thank you so very much, Hugh. I love getting these updates because, as you know, we follow these drugs very, very carefully throughout the years. And this is great news. Thank you so very much.
My pleasure, Ann.
So moving on, we're going to switch this up a little bit. I'd like to introduce Sarah Buckley, who is the Senior Director of Corporate Communications from CTI Biopharma. She is going to join us next. Sarah, are you there? Okay, great.
Yes. Can you see me?
Yes. Hi, thank you.
Hello. I'm just going to share my screen.
And Hugh, you need to, yeah, there you go.
And then let me put this into presenter view.
You need to unshare your screen. Thank you.
Didn't I do that? I'm sorry.
That's okay.
I can see your presentation, Sarah, not mine. So I think I'm.
No, but it's just that we can see you, Hugh.
Oh, you want me to turn off the camera? I'm sorry. Yeah, I don't blame you.
No, that's okay. You look great. But
All right. Thank you all. Can you see me, hear me, see the presentation from?
yes, everything looks great.
Thank you so much. So my name is Sarah Buckley. I'm a Senior Medical Director in Clinical Development at CTI. And we're developing pacritinib for patients with myelofibrosis, specifically for patients with cytopenic myelofibrosis who have thrombocytopenia and anemia that may make treatment with other agents challenging. As a disclaimer, pacritinib is an investigational product. It's not currently approved. Just some background on pacritinib. Pacritinib is a potent JAK2 inhibitor, but it has other potential mechanisms of action in myelofibrosis. The one that we're most interested in is inhibition of IRAK1. IRAK1 is a key component of the myddosome signaling complex, and so that's a necessary component for NF-kappa B activation via the canonical pathway. And so this pathway is upregulated in patients with myelofibrosis. It's considered to be a potential target for treatment in patients with myelofibrosis. And so this is there's a couple potential mechanisms of action that for pacritinib. Interestingly, it does not have any activity against JAK1 at physiologic dose concentrations, unlike other available JAK inhibitors. The big picture of pacritinib development is this. We've had two completed phase 3 studies called PERSIST-1 and PERSIST-2, and that was followed by a dose-finding study called PAC-203. And I'll briefly review all of those studies and some of our results. We currently have an ongoing phase 3 study called Pacifica that's specifically for patients with myelofibrosis who have severe thrombocytopenia or platelets less than 50. And talking about clinical development of pacritinib will not be complete without mentioning that we have submitted an NDA for pacritinib that's been accepted by the FDA for priority review, and they'll be giving feedback on that by the end of November. So we should be hearing fairly shortly whether pacritinib will be commercially available for use and in which population it would be available. So as I mentioned, there are two completed phase 3 studies already with pacritinib in patients with myelofibrosis, PERSIST-1 and PERSIST-2, and the schemas are shown here. PERSIST-1 was in first-line therapy, meaning patients would not have had prior exposure to JAK inhibitors, and patients were randomized between pacritinib and best available therapy. And at the time PERSIST-1 was started, that did not include ruxolitinib. PERSIST-2, on the other hand, looked at patients in first or second line, meaning patients who were naive to JAK inhibitors or who had prior JAK inhibitor exposure. And in those cases, patients were randomized between two different doses of pacritinib. It's 400 milligrams daily versus split into twice a day to see if that improved safety profiles. And best available therapy, which did include ruxolitinib, and almost half of the patients on the best available therapy arm received ruxolitinib on this study. And in both studies, patients were analyzed for spleen response as well as symptom benefit. So what we found is that in both of the trials, and you can look up each of the trials individually here. I'm showing pooled responses, but you can see that in terms of spleen response, patients who were treated with pacritinib had higher rates of spleen volume response compared to patients treated with best available therapy. And these rates were particularly high for patients treated with 200 milligrams twice a day. We saw these results overall and in patients with severe thrombocytopenia. This is a patient population that I'm calling out specifically because we see this as an area of unmet need, because patients with severe thrombocytopenia are often limited in what other medications are available to them and what other clinical trials are available to them as well. Similarly with the symptom score, this is measured by the total symptom score, or TSS. We were looking for TSS reductions of 50% or greater to determine response. And again, we saw that there are, in the overall population, more patients had a TSS response who were treated with pacritinib compared to best available therapy. And this was particularly true of patients treated with the 200 milligram twice a day dose. We saw this both overall and in patients with platelets less than 50, although the results in patients with severe thrombocytopenia were not statistically significant, likely because of smaller sample size compared to the overall population. We looked also at retrospectively at pacritinib use in patients who are ruxolitinib naive, so first line, based on the PERSIST-2 study. We looked specifically at PERSIST-2 because this is where the 200 milligram twice a day dose was studied, and that ends up being our recommended dose and the dose we're carrying forward. So you can get a sense of what responses look like in the first line with the recommended dose. The rate of spleen volume response is 28%, and the rate of TSS response was 33%, compared to lower response rates in best available therapy. Of note in this population, the majority of patients on best available therapy were treated with ruxolitinib. It was 32 out of 39. In both of these studies, one of the things we noticed is that there was stability in platelet counts and in hemoglobin counts over time, maybe even a slight rise, but it's what we're emphasizing is that platelet counts were stable. And this was true both overall and in patients with severe thrombocytopenia. Patients maintained stable counts while on full dose JAK inhibitor. Here we see on the prior slide, we saw platelet levels. Here we see mean hemoglobin levels over time, indicating stability. And after the PERSIST studies, there were some concerns over high-grade cardiac and bleeding events seen in some of the patients treated with pacritinib, particularly at the 400 milligram once daily dose. And so we did a dose-finding study. This was PAC-203, where we looked at patients who were refractory to or intolerant of prior ruxolitinib treatment, and patients were randomized across 3 doses. The highest dose was the 200 milligram twice a day dose that we looked at in PERSIST-2. And then we looked at lower doses all the way down to 100 milligrams once a day. And the goal of the study was to determine the optimal dose. And based on results from this study, including PK and PD analysis from all pooled studies, we selected the 200 milligram twice a day dose as the recommended dose for our phase 3 Pacifica study. But just to go a little bit into what happened on PAC-203, that study did not select for patients based on platelet counts. So patients could come in with any platelet counts. But interestingly, the median platelet count that we saw in our study was 60, so quite low, and with an interquartile range of 36 to 103. So just by selecting patients who were intolerant of or refractory to ruxolitinib, we ended up selecting for a patient population that was very cytopenic. So we see low platelet counts, low hemoglobin. Quite a few patients were transfusion dependent. There were, most patients had circulating blasts. So this was an advanced, heavily pretreated patient population. And within this population, we found that there were greater number of responses at the 200 milligram twice a day dose compared to lower doses. If you look at the top graph showing a waterfall plot of spleen volume change, you can see that there were more responses at the 200 milligram twice a day dose compared to lower doses. And one of the interesting things that we noticed is that most of these responses were in patients with baseline platelet counts of less than 50. In fact, of the patients evaluable for a spleen response who had platelet counts less than 50, you can see that there were 31% of patients who achieved the spleen response. We also asked patients how they felt after 6 months of treatment via this patient global impression of change, which is a question that says, compared to before you started this treatment, how are your symptoms right now? And you can see that the majority of patients, especially at the 200 milligram twice a day dose, reported feeling at least some improvement, with about a third of patients reporting feeling much improved or very much improved. Similar to prior studies, there was stability in hemoglobin trends and in platelet counts on PAC-203. So that's something that we've seen consistently across our studies. The adverse event profile on PAC-203 is notable, and I didn't show it for the prior studies because on PAC-203, we're looking at the dose that ends up being our recommended dose, and we put in place a number of guardrails and extra monitoring for patients in the event of any cardiac or bleeding events. And so we saw much lower rates of these events on PAC-203 compared to our prior studies. There was no trend in high-grade cardiac or bleeding events at the higher doses versus the lower doses on PAC-203. What we did notice is that patients did have some thrombocytopenia and anemia. Much of this was likely to be disease related. It was high grade because patients started out with high grade cytopenias. Other than cytopenias, the main adverse events that we saw with pacritinib were gastrointestinal, predominantly diarrhea, which was seen in 30% of patients. This was largely low grade and was manageable with over-the-counter anti-diarrheal medications. It generally resolves after about 2 weeks. So this does not persist for as long as pacritinib is taken. It starts after maybe a week or so and then goes on for about 2 weeks. And after that, patients don't generally experience it as a drug-related effect. And so that brings us to Pacifica. This is our ongoing phase 3 study in patients with myelofibrosis who have severe thrombocytopenia. The key eligibility criteria is having platelet counts less than 50. Otherwise, eligibility criteria are similar to our prior studies. Because spleen volume is a primary endpoint, patients have to have a palpable spleen. And because symptom reduction is a key secondary endpoint, patients have to be at least somewhat symptomatic going into the study. Patients can be JAK inhibitor naive or can have had a limited duration of prior JAK inhibitor exposure. And randomization is 2-to-1 between pacritinib at 200 milligrams BID or physician's choice therapy, which does include ruxolitinib. There's more information on Pacifica that can be found online at pacificatrial.com. Thank you.
Thank you very much, Sarah. That was great. We truly appreciate it. And I've been watching PAC for a lot of years, and so this is very good news. Thank you.
Thanks.
So moving on to Kartos Therapeutics, Nish Sayani will be joining us. He's the VP of European Medical Affairs. Hello again, Nish. Thank
Hello,
you for joining us today.
Not at all. Hopefully you can see my screen. Can I just check that's the case? And now I'm going to go into presentation mode. So if you just confirm that you can see everything.
All set. It looks great.
Perfect. Okay. Thank you, Anne. So good morning, good afternoon, everyone. My name is Nish Saini. So I work for Kartos Therapeutics in the Medical Affairs Group. And I want to thank Anne and MPN Advocacy and Education International for the opportunity to speak today and provide this brief update on Kartos Therapeutics and our program in myelofibrosis. So imagine many of you may not have heard of Kartos Therapeutics. We're a relatively small, privately owned biotech company based in the San Francisco Bay Area. And our focus really is the development of novel targeted treatments that have the potential to meaningfully improve the lives of patients with cancer. And we're currently developing an investigational agent called navtemadlin, previously known as KRT-232. And we believe this has potential to be a best-in-class MDM2 inhibitor. And what you see on the slide here reflects the current ongoing company-sponsored clinical development plan for navtemadlin across a range of indications, largely focused on the hematological malignancy space. And you can see here we have 4 studies, so a very big commitment to myelofibrosis. And just to summarize the studies we have ongoing in myelofibrosis, the one at the top you see here is a study, a phase 3 study, which is comparing navtemadlin to best available treatment in patients who are relapsed or refractory to JAK inhibitor treatment. And I'll give you a little bit more detail about that study in the upcoming slides. We also have a study looking at the combination of navtemadlin with ruxolitinib in patients who have a suboptimal response to ruxolitinib monotherapy. A third study is looking at the combination of navtemadlin with a novel cell trafficking agent that's being developed by our sister company, Telios Pharma. And this study is being conducted in patients who are relapsed, refractory, or intolerant to JAK inhibitor treatment. And finally, we have a window of opportunity study, which is looking at either navtemadlin or TL895, novel cell trafficking agent, in patients who are naive to JAK inhibitor treatment. So quite a lot going on in the MF space. I won't go into the details of the other studies we have in hematological malignancies, but you can see we're very active across a number of different indications. And of course, on this slide here, you can see the NCT numbers. So if you want to get more information of any of these studies, please do look on clinicaltrials.gov using those NCT numbers to identify the studies. So I wanted to provide a little bit more background to the mechanism of action of KRT-232 navtemadlin. I mentioned navtemadlin actually targets MDM2, and MDM2 is a key negative regulator of P53. And I'm sure many of you on the call today are very familiar with P53, which is often described as the guardian of the genome. And P53 actually responds to cellular stress induced by events such as DNA damage or oncogene activation to activate the transcription of various genes that are involved in either driving a cell into a rest or into apoptosis. Now, whether p53 drives a cell into arrest or apoptosis, we believe is quite context dependent. So if you have low levels of p53, cells go into arrest, whereas if you have high levels of p53, the cells are driven into apoptosis. The reason I explain that is because that forms the basis for the dosing strategy we are adopting for navtemadlin in our studies, which is to give the drug at relatively high doses for short periods of time to activate p53 to high levels and hopefully drive the malignant clone into apoptosis. So what you see on this slide is a little bit more information about how MDM2, this oncoprotein, how it modulates and regulates p53. And it actually does this by one of three different mechanisms. First, by binding to and blocking the p53 transcriptional activation domain. Second, by actually exporting p53 out of the nucleus into the cytosol. And third, by tagging or ubiquitinating p53 for proteasomal degradation. Now, MDM2 has been found to be abnormally upregulated in a number of different tumor types, including myelofibrosis. So the whole scientific hypothesis underpinning our studies is that by using an MDM inhibitor like navtemadlin, we can hopefully restore normal p53 tumor suppressor function and drive that malignant clone into apoptosis. And there's a little bit more detail on this slide about the relevance of targeting MDM2 specifically in myelofibrosis. I'm sure many of you are probably aware that one of the characteristic features of myelofibrosis is an elevated level of circulating CD34 positive cells. And it's this population that actually contains the malignant clone. And within these CD34 positive cells, there are an overexpression of MDM2, as you can see on the chart on the left-hand side of the slide here. So that overexpression forms the basis for targeting MDM2 in myelofibrosis. And then based on the mechanism of action that I've described for an MDM2 inhibitor, it's probably not surprising for you to learn that in order for the drug to be effective, in order for it to reactivate p53, you in the first place need an intact and functioning p53 protein. And what you can see from this graphic on the right-hand side here is that the incidence of mutant p53 in myelofibrosis is relatively low, less than 5%. So this high expression of MDM2 in CD34 cells and this low incidence of TP53 mutant protein, that forms the rationale and the basis for using a therapeutic strategy to target MDM2 in myelofibrosis. So as I mentioned earlier, we believe that navtemadlin has potential to be a best-in-class MDM2 inhibitor. And this belief comes from both the preclinical data that's been generated from the drug, but also the emerging clinical data across a number of different studies. And I'm going to share some of the data from myelofibrosis study with you in just a minute. But in terms of preclinical data, our studies have shown that navtemadlin is a highly potent and selective inhibitor of MDM2, and that's reflected by the strong target binding affinity and cellular potency that you see depicted on this slide. Now, this is an oral drug. It's rapidly absorbed by oral ingestion, and it's got a half-life that really lends itself nicely to once daily dosing. And because we believe navtemadlin can target the malignant CD34 clone, those cells that are overexpressing MDM2, we believe navtemadlin has the potential to deliver a disease-modifying effect in myelofibrosis. And what you can see on the right-hand side here is just a graphic illustrating the mechanism of action that I've already described. But essentially what you're seeing here is that navtemadlin essentially takes the brakes off p53 by inhibiting MDM2 and thereby allows p53 to drive apoptosis in malignant cells. So I just wanted to share some of the data that's already been generated for navtemadlin in a phase 2 study in myelofibrosis. And this study was initially presented at the EHA Congress in 2020. So here we go. This is the patient population that was recruited to this study was patients with relapsed or refractory myelofibrosis as defined by the criteria that you can see listed at the bottom of the slide here. I think many of you are aware that the prognosis for this patient population is pretty poor. Median overall survival has been reported in the range of 14 months for patients who've progressed on a JAK inhibitor. So a high unmet need patient population. One thing to point out is that we did not enroll to this study any patients who were JAK inhibitor intolerant or JAK inhibitor ineligible. This was purely a relapsed or refractory patient population. So this is a summary of the study schema from the presentation at EHA 2020. We recruited patients with TP53 wild type, primary or secondary MF, intermediate or high risk disease by DIPSS score. At EHA, we reported data from cohorts 1, 2, and 3 that you can see here depicted in the study schema. These 3 cohorts were exploring different doses and schedules of KRT-232. And the dose and schedule we ultimately decided to move forward with based on an aggregate view of efficacy and safety was cohort 3, which is looking at a dose of KRT-232 given at a dose of 240 milligrams once a day on days 1 to 7 of a 28-day treatment cycle. Oops, sorry. So just before I share some of the efficacy and the safety data with you, I just wanted to share some of the key baseline characteristics of the patients that were enrolled to the study. And the key take-home message here, I think, is that the baseline demographics you see on this slide really reflect a poor prognosis patient population that was enrolled to the study. The median age in the study was just under 70. The majority of patients, just under 70%, had primary MF. More than 70% of the patients had intermediate to or high risk disease by DIPSS score. Nearly 60% of them had the presence of high molecular risk mutations. And about 40% of patients were actually red blood cell transfusion dependent. Now, the spleen volume range at study entry was quite broad, but you can see here it ranged up to close to 8,000 cubic centimeters. So some patients with some very large spleens. What's important to highlight here is that actually we had 50% of patients who were on ruxolitinib at the time of their baseline scan at study entry. So we actually did not have a ruxolitinib washout period mandated in this study, and patients could have been on low dose ruxolitinib, either 5 milligrams or once or twice a day, up until the day before they started treatment with navtemadlin. And we think the fact that a lot of these patients were on ruxolitinib up until the day before they started treatment with the investigational agent may have actually confounded some of the spleen volume responses that we saw. And I will share a bit more detail behind that in the upcoming slide. So this is a summary of the key efficacy data that was presented at EHA 2020. And first of all, focusing on the left-hand slide here, what we're looking here at is the best spleen volume response. There were 25 patients who were evaluable for response at the time of the data cutoff, which was in April of 2020. And each bar in this chart represents an individual patient who was evaluable for spleen volume response at either week 12, week 24, or week 36. And you can see here, of these 25 patients, the vast majority of them, actually 16 of these 25, which is just about 65%, showed some improvement in spleen volume. And we actually had 4 patients here who achieved the SVR35 threshold. And that gave us a best spleen volume response of 16%. Now, what you'll see in this graphic here is that some of these patients are colored in brown, others in blue. The reason for that is these patients colored in brown were the patients who were on ruxolitinib at the time of their baseline scan, which could have been up to the day before they started treatment with navtemadlin. And what you can see, these patients in brown, none of them actually achieved the SVR35 threshold. And you can see visually their spleen volume reductions were not as good as those patients in blue who were off ruxolitinib at the time of their baseline scan. So what appears to have happened is these patients who were on ruxolitinib came off Rux the day before they started treatment. They then had some spleen flare and some Rux discontinuation syndrome, and that took some time to correct with continued treatment with navtemadlin. And so for that reason, moving forward, we're actually mandating a 28-day ruxolitinib washout period in our phase 3 study. And actually, if you remove these patients in brown from the analysis, the best spleen volume response changes from 16% to closer to 30%. What you see on the right-hand side here is the total symptom score improvement. This was available for 27 patients at the time of the data cutoff. 23 of these 27%, so that's just about 85%, showed some improvement in total symptom score. And you can see here a number of patients hit the TSS 50 threshold for a best TSS response rate of 30%. So this really was representing the first clinical proof of concept, highlighting the important role of MDM2 inhibition in the treatment of myelofibrosis. So I mentioned earlier that we think navtemadlin has the potential to deliver a disease-modifying effect. And this is actually reflected here in this slide, where you can see the ability of navtemadlin to rapidly and effectively reduce peripheral CD34 counts. And this is relevant because, as I mentioned earlier, it's this population of the CD34 cells that contains the malignant clone. And what you can see here is a nice dose response effect when you compare the 120 milligram dosing cohorts with the 240 milligram dosing cohorts. And you see a median of almost a 90% reduction in peripheral CD34 counts at week 12, which persist at week 24 with the 240 milligram cohorts. Now, we have a number of other pieces of correlative data that we're currently collecting, looking at changes in variant allele frequency, bone marrow fibrosis, changes in inflammatory cytokines, and I hope to be able to share that data with you at a later date. But again, we believe this emerging data continues to demonstrate the potential of navtemadlin to deliver a disease-modifying effect. In terms of the safety and tolerability of the drug, what you see here is the incidence of treatment emergent adverse events across the different dosing cohorts that were explored in the study. This is the dose and schedule that we'll be exploring in our phase 3 study. Now, the most common and frequent adverse events are really related to cytopenias or to GI adverse events. And it's important to state that we believe these are on-target effects as a result of MDM2 inhibition and P53 activation in rapidly dividing cells in both the bone marrow and in the GI tract. Important to state that these GI adverse events that you see here tend to be confined to the days of dosing of navtemadlin. So that's day 1 to 7 of a 28-day treatment cycle. You very infrequently see the adverse events persist outside of that range. And certainly we've been very successful in mitigating some of these GI adverse events with the use of prophylactic measures for nausea, vomiting, and diarrhea. We've also introduced a new formulation of the drug, which actually removes the requirement for patients to fast before and after taking the drug for 2 hours. And we believe that will also contribute to an improved GI tolerability profile moving forward. So just a couple more slides to share with you. And first, I just wanted to provide a little bit more detail about the BOREAS study. So that encouraging phase 2 data that I shared actually prompted the company to embark on this large phase 3 study called BOREAS. And this study is going to compare the efficacy and safety of KRT, of navtemadlin, I should say, versus best available therapy in myelofibrosis patients who are relapsed or refractory to JAK inhibitor treatment. We plan to enroll a total of 282 patients with TP53 wild type primary or secondary MF. The study will enroll across 130 sites in more than 25 countries, North America, Europe, rest of the world. And patients are going to be randomized in a ratio of 2:1 to either navtemadlin or to best available therapy. The primary endpoint of the study is going to be the rate of spleen volume reduction, and a key secondary endpoint is going to be TSS 50, total symptom score improvement. You can find more details about this study at on the website here. This is a website that we've created specifically for the BOREAS study, which provides more information on inclusion exclusion criteria and the sites that are open for the study. This slide just highlights the different countries that are going to be participating in the study. You can see it's a global study. In America, in the US, we have 14 sites, 12 of them are currently open, and activated for the study. So I hope you found that that quick summary, very helpful. We're a very small company, but I hope you can see from this we've got a strong commitment to the myelofibrosis space. And thank you again for the opportunity to speak today. If you would like to get more information about the studies or Kartos Therapeutics, you can go to either of these two websites. And of course, we have a Twitter feed as well, so please feel free to follow us on the Twitter handle Kartos Thera. So thank you very much, Anne.
Nish, thank
Yes.
you very much. That was extremely informative. Thank you. And we appreciate your time.
Sure, thank you very much.
Thank you, Nish. So we will move on to Sierra Oncology, and Carolyn Pack will be presenting. She is the Senior Director of Global Medical Affairs. Hello, Carolyn. How are you?
Good. How are you? Hi, Anne.
Thank you.
In one day.
Yes, we will someday in person. We will.
All right. Let me try to get my screen shared.
Okay.
Not sure which one it is.
While you're doing that, I'll mention that somebody did ask about the Mu variant, and as far as we know, the second or the booster shot should, be okay and help to ward that off, as well as the Delta variant. But the jury's still out because it's all so new to us and we just don't know. But our doctors still say, please get the booster shot if you can. Are you ready, Carolyn? I'm sorry, Caroline, and I'm going to exit.
I don't think, can you see my screen?
Sharing.
Can you see my screen?
Not
I
yet.
see I don't see your slides.
Okay, hold on one second. I think I'm frozen.
There we go.
Okay. It for some reason closed it. Okay, can you see my actual slide deck?
Yes.
Okay, let me put that in for
There you go. That's perfect.
All right. Okay. Sorry about that, guys. So good morning, good afternoon, wherever everybody is. Thank you, Anne, for the opportunity to present today. I am Caroline Pack, and I lead Global Scientific Communications for Medical Affairs at Sierra Oncology. We are a small company, just by way of background. We are a late stage biotech company and we're based in San Mateo, California. We all work virtually. We're a small company of about 100 employees or so, and we are on a mission to deliver targeted therapies that treat rare forms of cancer, including myelofibrosis, and that's what I'll be talking about today. We're committed to investigating new ways to change the cancer treatment paradigm, and we're extremely grateful to the cancer community and clinicians at large, because only together can we help transform that promise into patient impact. That's our mission. My main focus of the presentation today is momelotinib. It's an investigational agent in phase 3 development for the treatment of myelofibrosis. Momelotinib is an inhibitor of 3 key signaling pathways that contribute to the major manifestations of the disease. It inhibits JAK1, JAK2, and ACVR1 or ALK2, and it has the potential to improve the constitutional symptoms, splenomegaly, in addition to the anemia of myelofibrosis. Momelotinib is differentiated because of its potent inhibition of ACVR1. Elevated hepcidin levels inhibit iron transport and iron homeostasis, causing anemia of inflammation. And so inhibition of ACVR1 or ALK2 by momelotinib lowers those hepcidin levels and helps to improve that anemia, thereby reducing or eliminating the need for transfusions. And I just want to highlight, anemia is a significant unmet need for myelofibrosis patients. And although the currently approved JAK inhibitors can often improve the spleen and symptoms associated with the disease, they're intrinsically myelosuppressive. With momelotinib's unique mechanism of action and the low potential for myelosuppression, which supports higher dose intensity over sustained durations of dosing, and robust activity, irrespective of baseline platelet count, we believe that this differentiated clinical profile and then enables a very clear patient segmentation with the potential to benefit patients with anemia or thrombocytopenia or are at risk for developing them. Let me move on to my clinical development slide. So momelotinib has been extensively studied in 2 completed phase 3 trials, the Simplify-1 and the Simplify-2. Simplify-1 was conducted in JAK inhibitor naive patients in a head-to-head comparison with ruxolitinib. And Simplify-2 was conducted in anemic or thrombocytopenic patients randomized to either momelotinib or myelofibrosis best available therapy. This was not originally planned to be a head-to-head trial, but over 88% in the best available therapy arm, those patients were taking ruxolitinib when randomized to best supportive care. And the endpoints for both of these trials included measures of splenic response, symptom response, and transfusion independence at week 24. After which then all patients were then treated with momelotinib in an open label extension phase in each of the 2 trials. By way of results, and I'm not showing it here, but Simplify-1 showed that momelotinib's splenic response rate was not inferior to ruxolitinib. The symptom control appeared better with Rux than momelotinib, but the study was confounded by failure to stratify for symptoms, and there was an imbalance of higher baseline symptoms in the momelotinib group. Transfusion independence was better in the momelotinib arm. For Simplify-2, that showed that momelotinib was not superior to best available therapy for splenic response rate, but more patients receiving momelotinib had higher rates of symptom response and transfusion independence than in patients on best available therapy. We conducted several analyses on survival, and that was presented at ASH of last year. Long duration of survival were observed in both of those simplified studies. Median survival was longer than 4 years in the JAK inhibitor naive patients in Simplify-1, and approximately 3 years in those previously treated with a JAK inhibitor in the Simplify-2 study. And then further analyses were conducted and presented at EHA earlier this year, suggested that patients with myelofibrosis randomized to momelotinib who are transfusion independent at week 24 achieved a clinically relevant improvement in overall survival. Now moving on to our 2 ongoing clinical trials, our Momentum trial. I'll talk about that in a little bit more detail on the next slide. But I want to mention our ongoing expanded access program that has patient rollover from our phase 2, our Simplify-1, 2, and Momentum trials. I just also wanted to mention that some of our patients have been on momelotinib therapy for over 10 years, and when the Momentum trial is complete, nearly 1,000 patients will have received momelotinib. Moving on to our Momentum study design, we completed our enrollment of this phase 3 global study this past June, and we're currently awaiting top-line results. We expect to have that early next year. The study enrolled 195 out of planned 180 myelofibrosis symptomatic and anemic patients who were previously treated with a JAK inhibitor. There's going to be a washout period for 21 days, and then the patients were randomized 2-to-1 to either momelotinib 200 milligrams daily plus placebo, or danazol 600 milligrams daily plus placebo. At the week 24 mark, danazol patients will be allowed to cross over to the momelotinib 200 milligram once daily. The primary endpoint is TSS at week 24, and the secondary endpoints will be transfusion independence rate and spleen response rate also at week 24. And this momentum study aims to confirm the efficacy of momelotinib in myelofibrosis based on symptom, anemia, and splenic response rates. And assuming these data are positive, we plan to file an NDA in the second half of next year. And then new to our pipeline, we just added this. We made an announcement, a new compound to our pipeline, the addition of a novel BRD4 BET inhibitor, SRA 515, previously known as AZD 5153, is a differentiated BET inhibitor. It has high potency, it's very selective and known for its bivalent binding, which may offer more complete target inhibition relative to other monovalent binders. And our plan is to initiate a phase 2 trial with momelotinib and SRA 515 in the first half of next year for the treatment of myelofibrosis again. And we look forward to sharing more details about that study when we get closer to trial initiation. And then I get to my last slide, short and sweet. I just want to add to close that we're very grateful to the MPN Advocacy and Education International and the myelofibrosis advocacy community for all the work you're doing to raise awareness, to educate patients about this disease. And we appreciate the partnership we have with you. And I appreciate the opportunity to tell you a little bit about what we're doing at Sierra Oncology. Thank you very much.
Thank you very much, Caroline. This is exciting news by far. You know, it's just really good to know that this drug has stayed in the limelight and is doing so well. Thank you.
You're welcome.
So we will move on now to AbbVie and Brian Murray, who is a pharmacologist, and he will be presenting on AbbVie's behalf. And thank you for joining us again, Brian.
Thanks, Anne. Good to see you again. Can you hear me and can you see my slides?
Yes.
Fantastic. All right, I'm just going to switch this to presenter view.
There you go.
All right. Thanks, Anne. Thanks, everybody. Hello, my name is Brian Murray. I'm an Assistant Scientific Director in U.S. Medical Affairs with AbbVie. Today, I'm going to be talking to you about a novel agent that we're developing in myelofibrosis, navitoclax. But before I do that, I just want to thank Anne, Kathleen, and all of your colleagues at MPN Advocacy Education International for giving me yet another forum to discuss our program. We greatly appreciate the opportunity to discuss our trials. So we really appreciate that. Before I discuss our clinical program, I just want to review a few disclaimers. Navitoclax, the compound that I'll be discussing today, is an investigational new drug. It is not FDA approved in any hematological malignancy or any other indication. Safety and efficacy evaluation are ongoing. So the purpose today is to discuss 2 phase 3 studies with you. But before I do that, I think it would be good to give a little background on navitoclax. Now, I'd imagine almost all of you have heard of venetoclax, the BCL2 selective inhibitor in CLL and AML. Navitoclax was developed around the same time as a very close cousin. It inhibits both BCL2 and BCL-XL. BCL-XL and BCL2 are part of the same family of pro-survival proteins that are very important in the apoptosis cascade. Navitoclax in particular inhibits both BCL-XL, BCL2, and what we care about in myelofibrosis is BCL-XL. Now, you all know that myelofibrosis is driven by overactive JAK-STAT signaling. What you might not know is that JAK-STAT signaling also leads to transcription proliferation of BCL-XL and MCL1 pro-survival proteins. In the setting of JAK2 inhibition, in addition to the effects on myelofibrosis and the reduction in symptoms, you also see a decrease in the proliferation of BCL-XL. So what does this mean? These cells are now increasingly vulnerable to BCL-XL inhibitors such as navitoclax. Navitoclax inhibits BCL-XL, preventing it from sequestering the pro-death proteins like BAC, BAX, and BIM, allowing them to form oligomers in the outer membrane of the mitochondrion. And this makes it porous and releases factors that eventually recruit caspases, leading directly to apoptosis. So preclinically, we've shown that BCL-XL inhibition actually synergizes with JAK2 inhibition. We've also shown that BCL-XL inhibition can overcome JAK2 inhibitor resistance. On the basis of these findings and an additional preclinical model that we didn't publish, the company initiated a phase 2 study, a proof of concept study 4 years ago, which was eventually expanded for additional cohorts. Based on promising efficacy and safety data from the initial cohort of about 30 patients, which I will not be discussing today, but I would encourage you to check out the EHA 2021 or the EHA 2020 website to get updates on the data that we presented thus far. The company decided to move forward with 2 phase 3 studies. So that leads us to our first phase 3 study that I'll be discussing, which is Transform 1. This is a randomized, double-blind, placebo-controlled trial evaluating the combination of venetoclax and ruxolitinib in patients with primary and secondary myelofibrosis. So here's a simplified study schema. We're looking for 230 patients, and we're going to randomize them 1-to-1 into each of the treatment arms. So the experimental group, which I just mentioned, compared to a matching placebo and ruxolitinib control group. And then we'll treat them until disease progression, unacceptable toxicity, or death. We started activating sites in the study last fall, and we are continuing to do so. We are actively seeking patients for this study. And so I think now I'll talk a little bit about the sorts of patients that we're interested in for this study and the eligibility criteria to give you a sense of who we're looking for. So I'm not going to read through all of these eligibility criteria, but on the left, you can see some inclusion criteria, and I'll just point out a few highlights. So first off, as I'd mentioned, we're enrolling both primary and secondary myelofibrosis patients with myelofibrosis. We're looking for intermediate 2 or high-risk patients as defined by the DIPS Plus scoring system. Important to mention that the platelet cutoff for this study is 100,000 platelets per microliter. We also have some parameters around the symptoms related to the MPN symptom assessment form. And subjects have to have splenomegaly, defined as spleen palpation of at least 5 centimeters below the costal margin, or spleen volume of at least 450 cubic centimeters, as assessed by MRI or CT scan. In terms of our exclusion criteria, subjects cannot be candidates for stem cell transplant at study entry, and they're not allowed to go on transplant at any time during the study. They cannot have received prior treatment with a JAK2 inhibitor. This is subjects who have not received prior therapy with JAK2 inhibitors such as ruxolitinib. They can also not have received a prior treatment with a BH3 mimetic, again, a compound similar to navitoclax or a BET inhibitor. And patients cannot have leukemic transformation as evidenced by having at least 10% blasts in the peripheral blood or bone marrow. I won't read through the additional criteria, but you can refer to these slides, and I'll actually be referring you to a resource where you can find more information on the study and the next study I'll be discussing as well. So the next study I want to talk about is Transform 2. Unlike Transform 1, Transform 2 is our phase 3 study, which is open label, randomized with the same combination versus best available therapy in patients with relapsed refractory myelofibrosis. So Transform 1 was in those who are JAK2 inhibitor naive. This is in patients who have relapsed or progressed on ruxolitinib. So we're looking for 330 patients, again, randomized 1-to-1 to the navitoclax ruxolitinib combination arm or the control arm, which I'd mentioned was best available therapy. And there are a number of options here that are listed below in that blue box, and it's up to the investigator as to which therapy is most appropriate. But options include ruxolitinib, hydroxyurea, interferon, danazol, fedratinib, and again, continuing therapy until progression, unacceptable toxicity, or death. Eligibility criteria for Transform 2, very similar to Transform 1, with the notable difference that, again, because we require patients to have relapsed or refractory disease, there's additional parameters around that, which you can see in the lower left corner of the slide that I won't read through. Again, we're looking for intermediate 2 or high risk myelofibrosis patients. The platelet cutoff is 100,000, same as the previous study, and most of the other criteria outside of the progression are the same. I will note that only 1 prior JAK2 inhibitor is allowed, so patients cannot have received ruxolitinib and then fedratinib and then go on this study. It has to be, just the one agent. All right, so let's talk about some of the endpoints for each of these studies. Very standard for these sorts of trials. You've already seen them from some of the other panelists that have presented. The primary endpoint is spleen volume reduction at week 24, very common for these trials. The secondary endpoints, total symptom score reduction, various measures of survival, and we are looking at a bone marrow fibrosis reduction or improvement. In terms of our secondary endpoints, we are looking at various measures of quality of life, including fatigue, physical function, and others. And of course, we're assessing safety as always with our treatment emergent adverse events and our various clinical and laboratory parameters. If you want more information on both these studies, you can visit a publicly available website. It's abbeyresearchstudies.com. You'll find the same information that I presented here with some additional details on each of the studies, including some of the current locations for the sites that we've activated. There's a very handy Google map that's embedded into the website. You can find where the nearest location. If you have a patient that might be interested, potentially eligible for the study, and you can also connect with the Abbey research team there as well. There's a lot of good information there, so I would encourage you to check that out. That's all from me. Thanks again to Anne, MPN Advocacy Education International. Very much appreciate the time to discuss our clinical program today. Thank you.
Thank you so much, Brian. We really appreciate it. So we'll move on to Constellation, who Daniel Hennessy, who's the VP of Medical Affairs, will be joining us. Hi, Dan. I can't hear you.
Hello, Anne. Hi,
There
everybody.
you are. Hi. Okay.
Just getting my mic and my screen shared.
Okay. Thank you, Dan. We look forward to hearing from you.
All right. And please let me know when you're able to see my slides and if you can hear me okay.
We see everything and I hear you well.
Wonderful. Thank you very much. My name is Dan Hennessy. I'm the head of medical affairs at Constellation Pharmaceuticals. We are now a Morphosis company due to a recent acquisition that occurred in July. So we're very excited now to have a full portfolio with a focus on hematological malignancies. Our marketed compound, tafasitamab or Monjuvi, is currently approved for relapsed refractory diffuse large B-cell lymphoma. And in fact, we develop and commercialize with our partner Incyte. The focus today will be on our BET inhibitor pelabresib, which we'll be talking quite a bit about the MANIFEST program, but also the MANIFEST phase 3 trial. There's also compounds for immune-related indications and also CPI-0209, which is a second generation EZH2 inhibitor, another one of the epigenetic targeting therapies from the Constellation pipeline that's being explored in phase 2 in both solid and liquid tumors. Again, the focus of today's talk is to update on pelabresib and the mechanism of action and a brief update on the program. Pelabresib was designed to address the 4 hallmarks of myelofibrosis: spleen symptoms, anemia, and bone marrow fibrosis. Peg gave a nice introduction to BET and the proposed mechanism of action. We believe that epigenetic regulators have an impact on dysregulated gene expression that we see in myelofibrosis. And this is associated with pro-inflammatory cytokines, but also aberrant erythroid differentiation and megakaryocytic behavior as well. By targeting the BET protein with pelabresib, which is a potent and selective inhibitor of BET proteins, we believe that we have the ability to impact the cytokine production and the lead to the promotion of erythrocytic and megakaryocytic differentiation. Our phase 2 program, known as the MANIFEST trial, explored pelabresib in a multitude of contexts, from frontline therapy, the second line add-on approach, and also as monotherapy in relapse and ruxolitinib ineligible patients. And based upon the trial setting, we explored primary endpoints of spleen volume reduction, the ability to convert patients that were transfusion dependent to transfusion independent, and also symptoms and a whole host of exploratory endpoints that I will cover at a very cursory level today. This is a busy slide, but the key message here is that these are top-line results, primarily that we presented at ASH in 2020 across all three of the arms of the study. So again, this is the frontline context. This is the add-on context for those suboptimal ruxolitinib responses where we add on to ruxolitinib, and in the monotherapy context. And the key message is that we did see activity across the board on spleen and symptoms. And in addition, in those patients that were transfusion dependent at baseline, that were both in the arm 2, the suboptimal population, and also in the refractory population, we did see patients that were converting from transfusion dependence to transfusion independence, which was our primary endpoint for those cohorts within arms 1 and 2. In terms of our JAK-naive population, which was the arm 3, this slide depicts the primary endpoint and the key secondary endpoint of symptom response. So for the primary endpoint of spleen volume reduction, we used SVR35 at week 24, and based upon an interim analysis of 63 of roughly 85 patients that were mature at the time of the data cutoff in September of 2020, we observed 67% of patients achieving that SVR35, median of 50% reduction. And you'll notice from the waterfall plot that by and large, all patients, mostly all patients had a splenic response to the combination of an active therapy with an investigational therapy. In terms of symptom response as measured by TSS50 using the MFSAF, we observed 57% of patients achieving a symptom response as measured by TSS50 with a median of 59% reduction. Now, an exploratory endpoint in our JAK-naive, again, this is the JAK-naive combination population. On the left, this is not a comparative analysis, but by way of referencing the hemoglobin kinetics that were observed in the COMFORT-1 study. We also looked at the hemoglobin kinetics by measuring mean hemoglobin over time. And one potential hypothesis that can be derived from this is that the combination of pelabresib and ruxolitinib, we may have a more shallow dip in the anemia. We know that anemia pathogenesis is multifactorial, disease-related, treatment-related as well, and that may be mitigated. And on the right side, it may be more profound. I'd caution the small sample size, but this may be more profound in the patient population that is anemic at baseline. And I'll talk a little bit about some of our translational correlates that also feed in and support this potential hypothesis. From a safety standpoint, we see from many of my colleagues presenting today that there's a multitude of BET inhibitors that are being explored in myelofibrosis. And it's important to note that as a class effect, thrombocytopenia and GI toxicity are class effects that has been seen with a multitude of BET inhibitors that have been explored primarily in the solid tumor side, previous to exploration in the hematological space. In terms of pelabresib, the combination with ruxolitinib was generally well tolerated. Most common heme adverse events were anemia and thrombocytopenia. And the most common non-heme treatment emergent adverse events was diarrhea, but with also some nausea and abdominal pain. In this analysis, 78 patients in the ITT population, those were all low-grade GI toxicities. We did not observe any grade 3 or 4. 2 patients did discontinue pelabresib as a result of treatment emergent adverse events, and there were 2 grade 5 toxicities as well. Moving on to some of the translational exploratory endpoints. Interesting data here that looked at bone marrow fibrosis, but also some novel exploration of markers of erythroid differentiation and megakaryocytic, what we call histotopography. That's really how the megakaryocytes are clustering and behaving in a sense. We observed that 33% of patients in arm 3 receiving the combination of pelabresib and ruxolitinib had at least a 1 grade improvement in bone marrow fibrosis. And this tended to happen early in treatment. 88% of those occurred within 6 months of treatment. And again, this is at week 24. 2 patients did have worsening bone marrow fibrosis. And if I call your attention to the latter right-hand side, the CD71, which is this marker of erythroid differentiation, and CD61, which is a marker of megakaryocytes, what you observe here is that at week 24, this is a representative case where we saw increased staining for CD71, suggestive of more normalized hematopoiesis that we know is dysregulated, again, disease-related factors and also potentially treatment-related factors. And we saw an increase in this population in this exploratory analysis. On the other hand, with CD61, we observe, and this is a representative case, we observe a decrease in the clustering. We see a decrease in the staining of those megakaryocytes, and also see an increase. Those large white areas are fat cells, and these are indicative of potential for improvement of hematopoiesis and some of the other aspects of the pathophysiology of myelofibrosis that could be dysregulated and potentially ameliorated with this approach to modifying the gene expression. I think I covered all of the summary items. I'm going to move directly to talk about how we plan to validate this in our phase 3 trial. The focus of our program, the lead trial, is MANIFEST-2 trial, and this is a phase 3 trial. It has quite a bit of similarities to both TRANSFORM-1, the LIMBER study, and also to our arm 3, in that these are JAK-naive primary myelofibrosis or post-ETPV MF patients with advanced MF requiring therapy, splenomegaly symptoms. It will be stratified. We do include DIPS intermediate criteria 1, and that's actually one of the stratification criteria, 1 versus 2 versus higher platelets and spleen as well. Primary endpoint is SVR35 at week 24, and these patients would be randomly assigned to the pelabresib ruxolitinib versus ruxolitinib placebo. Also similarities, I know early on we had questions about the crossover, and very similar for patients assigned to the placebo arm that had 24 weeks of therapy. Meeting certain criteria are able to cross over to the investigational arm. In my last slide, this shows operationally, this is an international trial. We have sites that are active across the globe in Asia Pacific, Europe, and the US. We as well are also looking to increase the number of sites and really encourage the participation and accrual in the trial. If there's interest in learning more about the trial, we do have a website specific to the trial, www.manifestclinicaltrials.com. And we have an email address, manifest-2@constellationpharma.com. I want to again thank Anne and the MPN Advocacy and Educational International. It's really been an honor to be part of this, advancing a lot of really interesting science and clinical trials across the board. So really exciting time to be within myelofibrosis and advancing these clinical trials. I'll turn it back over to you, Anne.
Thank you so much, Dan. I really appreciate it. It's just so wonderful for me personally and for us to see these drugs doing so well and, becoming possibilities for our patient population who desperately need new drug therapies. And so, it's been wonderful for us. just to reiterate that this is being taped and are recorded, and a lot of our physicians that weren't able to join us today will be viewing this. And so, you know, this is a great clinical trial update for them, and hopefully for those of you that need patients in your trials, this will give them an opportunity to see what's happening and to, discuss that with their patients. We'll move on to Protagonist, and Nora Boyer is joining us. Are you with us, Nora?
I am.
Oh, you are? Hi, Nora.
Hi. So now I have let me see if I'm sharing here.
Your slide, but I'm going to take my video off and let that be.
Shall I, let's see.
There you go.
Here?
Yes.
Okay. Great.
Perfect.
All right. Great. So first of all, I'm not Paula O'Connor. Paula had an emergency and had to fly back to San Francisco. She was in New York at the US Open, so wasn't able to join us. So my name is Nora Boyer. I'm the Vice President of Medical Affairs. We have a few slides to go over, and let's see if I can go. I'm not able to advance it.
Oh, you should be able to from your end.
Yeah, I'm trying. Let's try it again. I'm gonna try it again. Sorry, it just.
Are you in presentation mode?
No.
Are you in presentation mode, Nora?
Yeah. I think so. I mean, I'm but I'm not able to. I don't know whether it's frozen. No. Where would I find that on the bottom? I thought I have share, new share. Would that be it?
It's sharing.
It says I'm sharing my slides.
Huh. That's the slide is there.
Yeah.
We can see that.
Mm.
Well, why don't you close it and open it again?
Okay. New share.
Sorry.
All right.
I'll tell you a little anecdote. When I was in New York visiting during the US Open, I, but of course was not attending, tried to make a restaurant reservation with some colleagues and could not. Nobody would let us in. And we called another restaurant that's pretty popular there. And I changed my last name to De Niro, and we were able to get in within minutes. It was quite funny and kind of shallow, but it worked.
Well, yeah, I just can't get this to do anything. Share, sound share.
Why don't we go on to the next person, Nora?
Okay. And
then we'll come back to you and see if you've been able to remedy that.
Great. That's a good idea. Thank you.
So we'll move on to Dr. Faye Feller, who's the Vice President of Clinical Development at Geron. Hi, Faye.
Hi, Anne. Can you hear me? Can you see me?
Yes, I can. I can see you loud and clear.
Great. Oh, am I too loud?
No, you're fine. So,
thank you.
Look forward to your presentation. Thank you.
Thank you, Anne. Give me a second to try to share.
There you go.
Okay.
Perfect.
Thanks. Good morning, everyone. And thank you again, Anne and MPN Advocacy, for having me here. I say this at every meeting, but I think it's incredible work that you do to bring together patients and providers and industry so that we can all learn from each other. And also, I think it's incredible the amount of development that's been done in in myelofibrosis within the last 10 years. It's just the research and clinical drug development has just exploded, and it's really encouraging for us on the drug development side and for patients and providers as well. So my name is Faye Feller. I'm head of the clinical development at Geron, as Anne mentioned. A little bit about us. We are a late stage biopharmaceutical company, and we have one drug that we are developing called imetelstat. So imetelstat is a first-in-class telomerase inhibitor. And our mission is really to improve the lives of patients with hematologic malignancies through the inhibition of telomerase. We were founded in the 1990s as with the mission to develop a telomerase inhibitor in order to treat malignancies. And over the course, we found imetelstat and to be most effective and best risk-benefit profile in the treatment of hematologic malignancies. So what is imetelstat? We are an oligonucleotide, so a first-in-class oligonucleotide for the treatment of hematologic malignancies that binds to the telomerase enzyme. You can see a little schematic of that here. Not allowing it to function in lengthening of telomeres. So we know that patients with myelofibrosis and in other hematologic malignancies, having short telomeres and high levels of telomerase activity, as well as expression of hTERT, the RNA template of the telomerase enzyme, portends for worse prognosis and more aggressive disease in some cases, and a shorter overall survival, especially in patients with myeloid malignancies. So by inhibiting telomerase, we hope to improve to provide clinical benefit to patients with these disorders. So we also know that in these malignancies, there's that the expression of telomerase is linked to the malignant cells. So malignant hematopoietic stem cells and the malignant precursor cells. So there is effectively targeting of the malignant cells by inhibition of telomerase while sparing and allowing for recovery of normal hematopoiesis. So we do have data in support of some disease modifying activity for imetelstat. Oh, now I'm having trouble advancing. Okay. Regarding myelofibrosis, most recently we had a phase 2 study conducted. This study was called MYF 2001 or EMBARK. And it involved a randomization for 2 doses of imetelstat 9.4 milligrams and 4.7 milligrams, looking primarily for activity in intermediate to or high-risk myelofibrosis patients who are relapsed or refractory to JAK inhibitor treatment. And as most of the presentations have demonstrated, everyone has their own spin on relapsed refractory. And in our case, we did not include any patients who are purely intolerant or ineligible for JAK inhibitor, but who really demonstrated progressive disease or after an initial response or just no response to prior ruxolitinib or any other JAK inhibitor treatment. So patients were then randomized to receive, after a washout period, either imetelstat at 4.7 milligrams per kilogram or 9.4 milligrams per kilogram every 3 weeks. Imetelstat is administered intravenously over a 2-hour infusion period. And the goal of the study was to look at the standard myelofibrosis co-primary endpoints of spleen response rate and symptom response rate. So this study was performed in exactly 107 patients. We ultimately determined that the 4.7 milligram per kilogram dose was not as effective as the 9.4 milligram per kilogram dose, and we will be moving forward in clinical studies with the 9.4 milligram per kilogram dose. And I'll show you here on the next slide a table of our results. So looking at the co-primary endpoints, we did not notice a significant spleen response. So only 10% of patients treated with the 9.4 milligram dose achieved a reduction in SVR by greater than 35%. However, if you look at the waterfall plot, many patients did have some spleen reduction. And again, these were patients who had previously responded and had significant improvement while treated with ruxolitinib or other JAK inhibitors. So in this patient population, it is questionable what degree of response is really clinically meaningful and has clinical meaningful benefit in terms of spleen reduction. We did note, however, a significant symptom response rate, so a 50% reduction in the TSS score in 32% of patients treated with 9.4 milligram per kilogram. Again, this was a relapsed refractory patient population, very large spleens, very symptomatic. So a third of patients were able to reduce their symptom burden by 50%. Most notably, however, we had a median overall survival of 28 months. And this was after a significant amount of time of follow-up. So we're very confident in the data of about a 28-month median overall survival, understanding that this is an uncontrolled study and it wasn't designed to look at overall survival. We still know that relapsed refractory patients or patients who discontinue a frontline JAK inhibitor treatment have a median overall survival of 11 to 16 months. So seeing this signal of 28 months was very provocative to us. In addition, there was noted a reduction in bone marrow fibrosis and mutant allele burden, specifically JAK2, CALR, or MIPL, in about 40% of patients. So this was a signal finding study, and we received, putting all this together, we do believe in disease modifying potential of imetelstat to change the natural history of the course of myelofibrosis. So I'm showing here the Kaplan-Meier curves for overall survival. Again, you're not meant to compare the 9.4 milligram per kilogram arm, which is in blue, to the 4.7 milligram per kilogram arm in red. But you can see our median overall survival of 28 months and 84% of patients surviving at 12 months, which is significant, which is about the median overall survival in the literature for patients who failed JAK inhibitor in the past. So with this information, we have currently opened a phase 3 study. So this is a unblinded, randomized, controlled study comparing imetelstat 9.4 milligram per kilogram to best available therapy in patients with refractory myelofibrosis. So these are patients who really have failed previous treatment with JAK inhibitor per strict criteria that's listed on the ct.gov website, and really intended for patients who would walk into their physician's office and would not be considered good candidates for further JAK inhibitor treatment. So with that in mind, the best available therapy arm does not include a treatment with a JAK inhibitor. Patients are randomized 2-to-1 imetelstat to BAT. Most notably, however, this study, our primary endpoint is overall survival. So we anticipate to randomize approximately 320 patients and look at overall survival with the spleen and symptom responses are key secondary endpoints. But we really want to see if what we saw during the phase 2 study of this potential for disease modification and extension of survival holds true in a randomized controlled setting. And we are the first study to look at overall survival in a phase 3 randomized manner as an endpoint. So that's all I have for today, but please visit geron.com or clinicaltrials.gov for more information. And again, thank you very much. This is informative both for myself and I hope for the providers who are online as well.
Yes, thank you very, very much, Faye. We really appreciate it. And again, this is a drug I've been following for years. And so it's nice to see the progress and very hopeful for the future. So we'll go back to protagonist and Nora Boyer if she is prepared or ready for us. What do you think, Nora?
I think I am. It does say presentation mode is checked. But while I was waiting, I got a notice saying that from our IT department that we're having a little bit of a slow with the connection. So I don't know if that was it or not, but let's try again.
Okay, let's try again. Thank you.
Okay. All right, let's see if so can you see my screen?
Very tight. There we go. Yes.
Yeah, okay, you see my screen. Okay, and now let's see if I can advance. I don't know. It won't let me advance, and I am in presentation mode.
Huh.
Definitely. It says use presenter view, and that's exactly what's checked.
Can you hit space, Nora, and see if that will work? Or the arrow key?
Yeah, I'm doing the arrow key.
Try
There.
me.
I got it. It worked. You there, Ann?
Yes, I don't
Okay.
I don't see the next slide, but you can.
Oh dear. Okay. Did you see my first slide?
Yeah, the cover is there.
Yeah, now I've gone to my second. It's up on my screen. And
It must be your IT glitch there because
I did send them to you. I don't know if it's possible for you to show them. Is it too late?
we can't do it.
Okay. Do you want me just to talk through them a little bit?
Sure, do you mind?
No, absolutely not.
We can share them later when we
Yeah.
put a link to them. Yeah,
And
sure.
we don't have very many slides. So,
That's
so
okay.
anyway, again, my name is Nora Boyer. I work for Protagonist Therapeutics, and we are a small biotech company in the Bay Area, Newark, California. We have about 100 employees, and our technology is based on peptide technology. And we're in different indications, but today I'm going to talk to you about our indication for hematology. Our lead compound is being studied in polycythemia vera. We're just finished our phase 2. It's called, the drug is rusfertide, formerly PTG 300. And it's a synthetic, rusfertide is a synthetic mimetic of the natural occurring hormone hepcidin, which is a regulator of iron homeostasis. So our drug is a copy, it's a mimetic. We've just finished our phase 2. We're fully enrolled and we're waiting for the data to mature. We actually are in discussions with the FDA to start our phase 3, which we are advising Wall Street should probably start in the first half of 2022. So that we expect to have more data. We had a presentation at EHA, and on I think 35 patients. We expect to have more, hopefully, in an upcoming hematology meeting at the end of this year. We are also looking at patients, looking to do we did a small study for patients with HH, and we're going to have that data available at the end of this quarter. So hopefully we'll start a larger study. We're also going to be starting another study for patients that have a hematocrit greater than 48. So those are the things. And then we'll also have an oral candidate being nominated. So all in the PV space. That was my slide. And let me just talk to you a little bit about our study. That was our proof of concept study. And so this is a study. We have 63 patients enrolled, fully enrolled. Data is maturing. The eligibility was they had to be phlebotomy dependent PV patients that were diagnosed according to the 2016 WHO criteria. They had to have 3 or more phlebotomies in 6 months with or without concurrent cytoreductive therapy. And they had to be able to receive rusfertide in the dose that was decided to be the correct dose for them subcutaneously on a weekly basis. So when they first started, the first part was a 28-week period, and that's where we would determine which was the correct dose of rusfertide for them. And then we would go into an efficacy evaluation period, titrating the dose. Then they would go into a blinded withdrawal, and that could be up to 12 weeks. And then the third part was 52 weeks where they were dosed and continued on. So the key endpoints were safety, maintaining hematocrit less than 45%, reduction in phlebotomies, and an improvement in symptom scores. So, as I said, we are in discussions with the agency and anticipate that we will start our phase 3 in the first half of 2022. We have received orphan drug status, breakthrough status, and a fast track, which is pretty exciting for us. And then my last slide here again is just sort of a summary of what I've said. The phase 2 enrollment is complete. We expect to share more details of the data at an upcoming meeting later this year, hopefully. We're talking with the FDA. We have those designations I mentioned. And an open label phase 2 is underway for patients that have a higher hematocrit of 48 or greater. And that's it. So short, and sweet.
I'm sorry that your slides didn't work, but that was a very good overview. And thank you so much, Nora. And we're very excited about Protagonist and this drug because I think it's greatly needed in this disease area. And so we appreciate it, and we appreciate the partnership.
Oh, great. Well, thank you. We're excited to just have to see what the data holds.
Yeah, yes. We'll keep our fingers crossed.
Right. Maybe next time we can go into the mechanism of action. We have some great slides on that.
Right.
Yeah.
That would be wonderful. Thank you again.
Thanks, Ian.
And so we'll move on to Pharmacentia and Dr. Ray, the head of clinical development and medical affairs. And Ray was with us yesterday as well as some of the others were, and for our patient presentations. And so Ray, thank you for joining us again and taking the time to do this. We really appreciate it.
Oh, you're welcome, Ann. Can you see my screen?
I sure can.
Thank you. So what I'll do over the next few minutes is just walk you through some of the advances we have done in next generation therapy for PV and ET, focusing on our lead asset, ropeg interferon alpha 2B, which I would just call ropeg just to make it a little bit easier. So ropeg, as the name implies, is an interferon. Interferons have been around for quite some time, and obviously being studied in various malignancies, getting FDA approval in '86 for hairy cell leukemia. And then you saw first reports of efficacy in ET and PV, as you can say, in the late '80s. Most, if not all of these studies were done with first generation or non-pegylated interferons. So you had issues with frequent dosing and tolerability issues, which as generations went on, these things were sort of remedied somewhat. And that's where ropeg was, why ropeg was developed and put into clinical trials to alleviate some of those issues. When you look at the mechanism of action of interferons, it's a rather complex story. It depends on the cell type you're looking at, as well as the microenvironment upon which it is acting. This is just a schematic showing you the degree of complexity of where interferons actually work and which cell types they work on. And then when you just put this into some sort of sentences, you can see some of the activities, major activities that you've seen from the interferons, like stimulating cytotoxic T cells and NK cells, induction or activation of pro-apoptotic genes, and that may be more relevant to when we're looking at PV and ET. And angiogenic activity. So I won't get into all this, but I said it's a complex molecule that has complex mechanisms of action depending on the cell type, as well as the microenvironment upon which it's acting. So the advances, this is a schematic of ropeg interferon alpha 2B, as I mentioned previously. It's a third generation pegylated interferon. By that, I mean the second generations have polypegylated 8 to 14 different isomers. Ropeg actually is a single isomer based on a novel technology of adding a proline to a native interferon and then pegylating it specifically on that proline residue. So you wind up with almost exclusively a monopegylated molecule. This is very important because it leads to improved PK/PD sort of characteristics, which allows for better tolerability and less frequent dosing, as well as more exposure. And we can see that in some of the efficacy data I'll show you shortly. This was the clinical development program that was run for ropeg in PV. Basically two main studies, the PEG and VERA study, which was a 51 patient, dose ranging study looking at the MTD. Of note is the median duration of observation was 5.1 years. So we followed these patients out well past the initial portion of the study. And then there was the PROUD PV continuation PV, which is abbreviated here as Conti PV. And this was our pre-planned open label single arm study. And you can see it there. And we have followed these patients past 5 years or 60 months. Some of this data was presented by Dr. Gissinger at the previous ASH meeting, and I'll show you some of that data. So let me walk you through a little bit on the PEG and VERA data, and then I'll show you some data on the PROUD and Conti. These were the primary endpoints, as I mentioned, looking for an MTD. Those of us who, probably everybody on the phone who's been involved with biologics know that there's really no MTD for a lot of these drugs, and the FDA is actually going through the exercise of having us focus on dose optimization versus using the older chemotherapeutic approach of looking for MTDs. But that's where we ran the study. I'll show you the dosing schema, but that's the dosing range showed on the left, and secondary endpoints were your typical safety, tolerability, as well as some efficacy and PK parameters. This is a schematic about the dosing. So it started incredibly low at 50 micrograms Q2 weeks, and then was elevated to 100, 150, 225, 300, and you can see the rest there. So a rather slow titration with relatively low doses. No MTD was observed, so 540 micrograms was quote unquote listed as the MTD, but there actually was no dose limiting toxicities observed during this dose ranging study. Of note, as I said, we followed these patients out past 5 years. A great majority of the patients after approximately 94 weeks were put on Q4 week dosing as opposed to Q2 week dosing. And the hematologic response and molecular responses stayed stable during this time, possibly allowing for patients after they've been on the drug for a while and their count stabilized to go on a monthly dosing regimen versus a Q2 week dosing regimen, which obviously would be very convenient for the patient. This is just looking at the CHR, or the complete hematologic response. It's defined below on this slide. I won't reiterate it. And we looked at CHR and CHR with normal spleen size. In this particular patient population, because they were relatively new in their disease, if I could use that phrase. So the spleen was really not enlarged nor an issue in these patients. But because of FDA requirements and our conversations with them, we measured it anyway. And you can see the overall response rate, 80.4% of patients achieved a complete hematologic response at some point during the duration of the trial. So then when I switch gears and I'll talk about the PROUD continuation study, the slide I showed previously showed you the top portion of this, which was the pre-planned single arm analysis looking at patients. This one has 48 months of ongoing treatment, and then plus the 12 months, we have 5 years of data, which, as I said, was presented. In the initial first 12 months, we actually had a comparative versus hydroxyurea, and I'll show you some of that data subsequently. But just make of note, the exploratory analysis of the HU BAT arm was actually added post hoc, and that's why there was a study entry gap of around 21 weeks, because they were trying to operationalize the study, that particular arm. So there was a bit of a delay in getting patients rolled up into it. So you look at this complete hematologic response data, and we have 3 different categories here of CHR, and then no increase in spleen size and normal spleen size. Again, this was just an FDA requirement. And you can see in all patients at 12, 24, and 36 months, the complete hematologic response rate goes at 12 months from around 56, 57%, and stays above around 70% going outward. When you look at the overall response rate, which basically is anybody who achieved the CHR at any point during the observation period, you could see, as I showed on the previous slide, it's with the PEG and VERA data of 80.4%. This is actually 84.3%. So the effect size is actually quite consistent across these two studies. You can see the two other overall response rate variables on the graph on the right. So of particular interest to us, given that ROPEG and interferon, ROPEG interferon tends to, we believe, work at the level of the bone marrow and the JAK2 mutated hematopoietic stem cell. We looked at the allelic burden. I'm sure you know what that is. In this particular instance, it's the percentage of mutated JAK2 alleles over the total. This is ROPEG versus the HU-BAT. And as you can see, at baseline, they're fairly consistent. Both arms tend to decrease within the first 12 to 24 months. And then, not surprisingly, given that hydroxyurea is a non-specific myelosuppressive, the allelic burden in the HU-BAT arm goes up almost to above baseline, whereas the ROPEG arm allelic burden continues to drop. Of particular note is we had a complete hematologic response in around 14% of patients, which basically at the 4-year time point, which basically means there was no measurable residual disease at that point in time, which is quite significant. And again, just feeds back to the mechanism of action and the thought that the ROPEG actually preferentially and specifically kills the JAK2 mutated hematopoietic stem cell, which is driving this disease. This is actually Kissinger data from he presented at ASH last year, and it looks at the median JAK2 allele burden, and this is using the LOCF methodology. And as you can see in the ROPEG arm, the allelic burden drops significantly from around 35%, which I showed previously, and it's going down to almost between 5 and 10%. Some investigators actually believe that at this point in time, you may be able to employ a drug holiday, but that's to be determined. And again, in the control group, you can see that initially goes down, and then after 12 months, it starts going up again and actually rising above baseline. The mechanism behind this is as yet to be understood. So when you take the allelic burden and then we quantify it in what's called the molecular response or partial or complete molecular response, and that's defined on the bottom left. And again, this data published in the Lancet Hematology. You can see that the molecular response for ROPEG actually improved over time between 12 and 36 months. So basically going from a molecular response of 44% at month 12 to around 66%, excuse me, at month 36. And it was fairly consistent with what happened at month 24 or year 2. When we look at the molecular response, this is basically mirroring what I just showed you with the allelic burden, since the two of them, those variables are actually related. You could see that with the ROPEG arm, the molecular response goes up rather significantly, stays around 70%, whereas in the control arm, or the hydroxyurea BAT arm, the molecular response actually drops. So again, showing the specific effect of ROPEG on the JAK2 mutated cells that are driving this disease. This is the adverse events. We didn't see anything out of the ordinary or unexpected. Was some elevations in ALT and GGT, but these actually were reversible. So now let me change gears a little bit and I'll talk to you about essential thrombocythemia. This is actually a study that is currently ongoing. It's called the SURPASS-ET trial. We were trying to focus on four specific items, looking at outcomes, a deep and durable hematologic response, looking at a drug that's safe, again, low toxicity, improved tolerability. These patients tend to be on the drug a long time. Looking at less frequent dosing, and again, I showed you the data from the PEG and VERA trial when many patients were switched to Q4. And then looking at disease modification, again, I showed you data related to allelic burden and molecular response, which certainly indicates that ROPEG does actually modify the disease and not just treat a sign or a symptom of it. The study goal was to evaluate the safety and efficacy of ROPEG. It's an phase 3 open label randomized active control. You can see the timing there of 14 months for patients that are entering into the trial. The patient population is basically hydroxyurea resistant resistant patients. And we have approximately 60 centers. We are just opening up sites in Canada and Europe. We have had sites open in the US for about a year: Japan, Taiwan, China, South Korea, and Hong Kong. It's about 160 participants. We're currently at about 80, but we need 160 to have 130 to complete the study, and we're sort of on track for that. This is the study diagram. Just a couple of points where we have the stratification factor of country, US, non-US, that will be modified based on the addition of Canada and Europe. But the more important part, it's a parallel group study looking at HU resistant patients either getting Ropeg Q2 week, at least initially, versus anagrelide. And you can see the schema here. It's a 15-week treatment cycle with a 4-week follow-up. These are the main inclusion criteria. Again, people can read this at their leisure, but it is high-risk ET patients, and we see how they define the high risk. Main exclusion criteria are the typical contraindications or any comorbidity that might hinder their participation in the study. The primary endpoint is the response to treatment by the modified ELN criteria, and it's all listed there on the left as well. And we're also looking at total symptom score, which I believe several presenters actually went over the TSS during their presentation. Secondary endpoints, we're looking at durability of response, thromboembolic events, et cetera. And also looking at several of the genomics going with changes in CALR, MIPL, or JAK2 over time. We do encourage people to reach out to us. We do have the trial listed on clinicaltrials.gov. You can see the NCT number there. We also have a microsite, and you can see it's surpasset.com. So we welcome any interest in the study. We do also have available study information for patients and healthcare providers, and we can provide this also via our website as well as via mail for our medical information portal. And last but not least, PharmaSense is also looking into a host of other disease states besides the hematologic malignancies of PV and ET, looking at a next generation P1101, which is actually the previous name for Ropeg. We're also looking into hepatocellular carcinoma and some of the other solid tumors, as well as some dermatologic diseases as well. So Anne, I covered a lot very quickly, so I hope that was helpful.
You did a great job, and thank you very much. And I know that many patients that are on that have done very well on interferon are anxious to see what happens with Ropeg. So thanks, Ray. We really appreciate it and appreciate the partnerships with all of you. As I said at the onset of this program, we could not do it without your help. To bring the resources and the events to our patients in the entire MPN community. So thank you very much for your time. And we will get this information to those physicians that weren't able to attend. And hopefully it will spark some interest and get some information to their patients. So again, thank you. Be safe. Get a booster shot, get vaccinated if you haven't, and take care. Hope to see you all in person soon. Bye now.
Thank you, Anne.
Thank you.
Thank you, Anne. Bye, everyone.
Bye.
Bye, Anne.
Bye.